The sympathetic nervous system is widely known as the body's panic button, the fight or flight response. But clinically, its primary function is mechanical. It is the core biological engine that forces blood into vital tissues to ensure survival under stress. Every single sympathetic signal that drives this perfusion originates entirely within the central nervous system. Specifically, the wiring begins deep inside the spinal cord, anchored in a structure called the intermediolateral cell column. On this diagram, the column doesn't run the full spine. It's confined strictly from T1 to L2. Pulling back, an architectural problem emerges. How does this single, constrained origin successfully command dozens of contradictory physiological responses simultaneously? The system achieves this through a dual delivery architecture. It utilizes highly targeted point-to-point neural networks alongside massive whole-body endocrine broadcasts. To understand the sympathetic response, we have to stop looking for a single distress signal. We must trace the physical wiring, through its chemical manufacturing steps, down to localized cellular receptors. The outward physical wiring of the sympathetic nervous system relies on a universal protocol, a strict two-neuron chain. The first link in this chain is the preganglionic neuron. Structurally, it is always short. To cross the gap to the next nerve, it always releases the neurotransmitter acetylcholine. At this relay station, that acetylcholine binds to a very specific target, nicotinic NN receptors, located on the receiving end of the postganglionic neuron. This second nerve, the postganglionic neuron, is structurally long. It reaches out to the target tissue, and in the vast majority of sympathetic pathways, it releases norepinephrine as its localized neurotransmitter. There is one major clinical exception to this two-neuron rule, and it happens at the eccrine sweat glands. The sweat glands are firmly wired into the sympathetic nervous system. However, their specific postganglionic neurons do not release norepinephrine. They release acetylcholine instead. This terminal acetylcholine targets muscarinic M3 receptors directly on the sweat glands, triggering the production of sweat. The origin in the spine doesn't dictate what the target tissue does. The specific neurotransmitter released at the very end of the line determines the local response. This brings us to the biochemical foundation of the system. This section outlines the catecholamine ladder, showing how these localized signals are manufactured. It begins with the base amino acid tyrosine, which converts into DOPA. DOPA is then converted into dopamine. This is the first active signaling molecule produced on the ladder. Dopamine is subsequently converted into norepinephrine. For standard sympathetic nerve endings, this is where the process stops. But there is a final potential step on the ladder, the conversion of norepinephrine into epinephrine. This final conversion is only possible in the presence of the enzyme PNMT. Standard nerve endings simply do not have PNMT. It is located almost exclusively within the chromaffin cells of the adrenal medulla. The presence or absence of this single enzyme dictates the entire delivery method. Without it, you get a highly localized neural network running on norepinephrine. With it, you generate a systemic hormonal network driven by epinephrine. This table of receptors and body effects illustrates how the specific G protein receptor dictates the end organ response. Take the alpha-1 receptor. When stimulated, it triggers the GQ signal pathway, causing a sharp release of intracellular calcium. Clinically, this calcium spike causes profound vasoconstriction, pushing up mean arterial pressure, while simultaneously causing pupillary dilation in the eye. This specific pathway is why we use norepinephrine drips to manage patients in septic shock. Shift over to the beta-1 receptor. This one triggers a different pathway, GS, which increases intracellular CAMP and calcium. Because of its location, beta-1 activation directly stimulates the heart, increasing both heart rate and contractility. It also hits the kidneys, triggering the release of renin to raise blood pressure. Then we have the beta-2 receptor. It also utilizes the GS pathway to increase CAMP, but here it activates pKa. The result is the exact opposite of alpha-1. Beta-2 triggers active vasodilation in skeletal muscle and forces bronchodilation in the lungs, which is exactly why albuterol acts on this specific receptor to stop asthma attacks. It also drives intracellular potassium shifts. Finally, the alpha-2 receptor acts as the system's internal brake. It sits on the presynaptic sympathetic terminals themselves to create a negative feedback loop. When stimulated, alpha-2 actively decreases local norepinephrine release. which lowers overall blood pressure. This makes it the exact pharmacological target for drugs like clonidine. By distributing these distinct receptor types across different organs, the body ensures that a massive, system-wide chemical release translates into highly customized, tissue-specific survival mechanics. Receptor activation is also dose-dependent. We see this most clearly in the clinical application of dopamine. At a low dose, dopamine almost exclusively targets D1 and D2 receptors, specifically causing renal and mesenteric vasodilation. Push to a medium dose, and the chemical concentration causes a crossover effect. It begins activating beta-1 receptors, actively stimulating the heart. At a high dose, the sheer volume of circulating dopamine overwhelms the alpha-1 receptors, resulting in massive systemic vasoconstriction. This principle of overwhelming volume transitions us away from targeted local pharmacology and directly into the role of the adrenal medulla. Looking back at the diagram, you can see the adrenal medulla is essentially a modified sympathetic ganglion. The short preganglionic nerve releases acetylcholine directly onto the adrenal chromaffin cells, completely bypassing the need for a long postganglionic neuron. Because these chromaffin cells contain the PNMT enzyme we discussed earlier, the gland instantly dumps epinephrine directly into the fast-flowing bloodstream as a hormone. The adrenal medulla allows the sympathetic nervous system to bypass the physical limitations of point-to-point neural wiring. It creates a broadcast signal, instantly saturating every accessible receptor in the body during an extreme emergency. The body achieves simultaneous contradictory goals, like constricting skin vessels while opening lung airways, through a sophisticated layering of anatomy and biochemistry. A unified spinal origin from TIM1 to L2 provides the signal, while the specific G protein receptor at the tissue level determines the actual physical outcome. It operates across a strict divide between the localized precision targeting of norepinephrine synapses and the blunt whole-body saturation of epinephrine from the adrenal gland. In clinical practice, medical professionals do not simply treat a vague fight-or-flight response. They actively manipulate this precise circuitry to dictate tissue perfusion. They select a targeted alpha-1 norepinephrine squeeze to stabilize collapsing vascular tone in a septic patient, or they utilize a broad-spectrum epinephrine flood to restart a stopped heart during cardiac arrest. Mastering this sympathetic architecture, understanding exactly which chemical binds to which receptor, is the fundamental toolset required to keep dying patients alive.